So I'd like to first of all welcome Dr. Jeremy Levan. Dr. Jeremy Levan serves as Chairman and CEO of Ovid, focusing on developing innovative medicines for epilepsies and seizure-related disorders. Previously, Jeremy held the position of President and CEO at Teva Pharmaceutical. Apologies, I'm going to mispronounce a few things, the difference between British and American accents. And he was also part of the Executive Committee at BMS, as well as the Global Head of Strategic Alliances at Novartis. He now presides as Chairman of the Board of TIA and participates on the Board of Directors for BIO, where he recently served as Chairman. In 2023, Jeremy received the Scrip Lifetime Achievement Award, was recognised by Endpoints in 2021 as one of the 60 living pioneers in the industry, and was named one of the 25 most influential biotechnology leaders by Fierce Biotech, and was listed among the Pharma Voice 100 CEOs in 2020. Jeremy completed his BA, MA and DPhil at Oxford University, so it's lovely to have a speaker with UK roots, and he obtained an MB BCIHR from Cambridge University. So that is Jeremy, our first speaker, and moving on next to Dr. Amy Mathia, who serves as the Chief Medical Officer at Graviton Bioscience Corporation, bringing with her a profound depth of clinical and academic experience acquired over nearly 20 decades. That would be a very long time, wouldn't it? Her recent role as SVP of Clinical Research at Equilib Biopharmaceuticals reflects a commitment to medical innovation, following an illustrious tenure in academia. Amy works as Associate Professor at MT Sinai and NYU, and is notable for advancing PET and MRI applications in breast cancer. A recognised leader, she achieved fellowship in her Society of Breast Imaging and played a pivotal role in shaping breast imaging curricula. Her mentorship, alongside contributions to over 30 peer-reviewed articles, underscores her influence in radiology. Her academic foundation includes a distinguished BS from the University of Wisconsin and an MD from UCLA, augmented by her Howard Hughes Fellowship at the NIH. Her career is marked by passion for translating complex science into tangible medical progress. Wow, that's impressive, you two. We're very lucky to have you here today, and you are going to be speaking on the topic of ROCK2 inhibitors. And just to those joining today, the reason we've asked this talk to take place is our vision is to find a cure for Cavernoma. And our mantra for this year and for Cavernoma awareness is hope for a cure. And ROCK2 inhibitors is one of those hopes. So what I'm going to do now is I'm going to begin screen sharing, and Jeremy, I'll have control of that for you. We'll take you through that, and then I will stop sharing and hand over to Amy, as I understand she's got some slides to share directly. So if you want to bear with me for 1 minute, I'm just going to share. I'll put it up from the beginning, and I'm going to skip through this because we've gone through this bit here. And I'm going to begin, Jeremy, handing it over to you, and let me just spotlight you now so everybody can see you. Jeremy, you've now got the spotlight, and I will move as you direct me.
Great. Thank you so much, Helen. And you forgot a key piece for those in the UK that I think is important. I was at Holland Park Comprehensive. That's where I did my GCEs and was hopeless. I got 2 Cs and 2 Ds. And it's a testament to those with the vision that they allowed me into a university to go on and do my degrees. And I showed them because one thing drives everything you do, and that's passion. And for me, there's only one thing that matters, and that's finding medicines that can help people, myself, others. But most importantly, every time you find a medicine, the idea is, and the concept is, that you'll change your life. And what I'd like to do really is to talk to you now about that, because I think we have a chance of changing lives, and we'll do that. So if I may I get the next slide, please? One of the most important things as a public entity is just for you to understand that we are a public entity. And so whatever I say here, you need to take in that context. But let's leave the legality out of this for the moment. and concentrate on the meat and potatoes of what we will be talking about. Next slide, please. I want to start by saying Graviton, I'll talk to you about Graviton, and then I'll talk to you about Ovid. But before I even go there, let me just say everything we do together is in concert with the community. I've come to understand, and I know the people in Graviton understand, that without a community, without the people giving us their ideas, thinking us through problems, establishing for us what is important, we simply cannot develop a medicine. And I have to say I was inspired by Rob Rowe, Steve Kern, and Alex Ford, and the others in the previous panel, because listening to them brings it right back to the facts of the matter. Well, let me tell you about Graviton now. Graviton is a very remarkable company. It was founded by those who understand what ROCK2 inhibition means. Now, ROCK2 is a protein. It's a protein that actually has a function within a cell, and that function is intimately related to the disorder of Cavernoma. And so Graviton's CEO, Dr. Sam Waxel, for years worked on this area. He knows it, he understands it, and indeed has already developed a medicine in a related area targeting this called Rezurock, which is helping and benefiting many patients. It's a remarkable achievement. Actually, it's one of 11 different medicines that he has discovered, and I believe he's the person who's discovered more medicine than anybody else in the industry. So Graviton has solid experience. Amy, who will talk to you shortly, leads in a remarkable effort, and we're very proud to be working with them. They have decades of information, decades of knowledge, decades of experience in this area. So if anybody can crack the science, they can. Ovid didn't start in that area. We started with the principle that it's our job to develop medicines for rare brain disorders. Frankly, that means you have to start by asking people, what does it mean? What does that disorder need? Where can we do something? Our whole approach is to find small molecules. Those are things that you can take as a pill or can be injected, but primarily as a pill. And to use that capability to develop new medicines. The team has a great track record of doing this. And the one thing that we absolutely insist on, and Helen, I'm very grateful for the manner in which you worked with us, is to actually work with the community, because we start there before we ever start to look for a compound or a medicine. We start by asking, what does the community need? Next slide, please. So we've brought together two great skill sets. Ours from the development of medicines, Graviton from a deep understanding of the science. And we're trying to catch Graviton up. I always like that, but frankly, I've never yet been able to second guess them. I try. I can't, because basically I'm a technology geek. I love understanding science. I know they love understanding how to develop a medicine. But most importantly, we jointly love working with communities. So when you bring us together with their expertise in ROCK inhibition, we know what the right molecule at the right time, and that is Graviton's 101, and as we call it, OV888. We are sure that this has the potential to target CCM's fundamental root causes. And this partnership brings together two small companies with the community. So really it's a troika. By bringing us together, we are force multipliers. Next slide, please. Well, it's important that you know who you're dealing with. And I don't want to underestimate that importance, because every company is built of people. And if you don't like those people, you won't work with them. I know that. That's the way I've always worked. I've always tried to understand who are the people I work with in the community. Can you trust them? Will they be sensitive to us? Will they understand what my child needs? Every single person on this in these two companies does that. And every single person here is devoted to finding the right medicine. Sam Waxel, as you see there, Amy, Alexander, Ruwu, Pushpam Bathroy. These are phenomenal people. And each one of us, for better or for worse, we've been through the ringer at many different companies. Sometimes I can tell you I did not like it, but I learned a ton. And we were very successful in doing so. On the other side, on the Ovid side, I'd want to bring to your attention our team. Julia specifically has developed many drugs and have taken them into the community. And Raquel is devoted to actually ensuring that we are glued and hear everything that the community wants us to hear. So this is a very, very strong team, and one that we hope will work with you for many years to come. Next slide, please. Now, as I said to you, that's our internal team. We work incredibly closely with the community. Helen, I salute you. I heard that you're an army of one, but actually around you, you have an army of hundreds. And it's your ability to channel their power in the United Kingdom, and then to work with Connie also in the United States and Issam, which will actually make a difference. The beauty of this capability that you have is that your arms and your influence stretches around the world. I saw somebody joined a few minutes ago from Sweden. I'm not surprised. They need to hear the voice of Cavernoma, and you're it. And I'm very proud to be working with you, Connie and Issam. What a great delight for us. Frankly, every single one of you makes contributions. And if we can link ourselves and strengthen our ability to work with you, I'm sure that the power that we create will be far more than the sum of the parts. And let me take the next slide. Now, I know this next slide is just a picture, but in my mind, I'd like you to think about this a little bit. A picture is 1,000 words. When I did my training at the Hammersmith Hospital and then subsequently worked in liver transplant, we were sent down to King's College because that was the center. Actually, I knew something very special about it. I knew that it had other areas, but at the time I wasn't able to work in them. Now I can. So Helen, thank you so much for taking me and Meg down to King's. It was an eye-opener, actually, of course, because I'm now a little older than I was. There's been a huge expansion, and it's fabulous. And we got to meet the remarkable Mr. Daniel Welsh, utterly committed to this area. And so I'm just phenomenally, let me underline that word, phenomenally impressed by what you've accomplished. And I'll go back to that moment at Holland Park and those 2 C's and 2 D's. It's all about passion. It's all about focus. It's all about actually understanding what people need. And if you do that, you can accomplish so much. And we're going to accomplish a lot with you. Thank you so much. And I'm now going to hand over to Amy.
So Amy, I've now stopped screen sharing. So it's over to you to screen share, and I'm going to turn the spotlight off, Jeremy, and then put the spotlight on you. So give me 2 seconds and I'll just do that now. Lovely. Amy, you're now spotlighted, so it's over to you.
Oh, excellent. Can you hear me just fine?
And I can hear you nice and clearly.
All right. Well, hello, Helen. Thanks very much for having Jeremy and me here today. We really appreciate the chance to get to know your community. And I've been introduced already. So hello, I'm Amy. I'm the Chief Medical Officer at Graviton, and I'm going to talk about the science behind the Graviton and Ovid collaboration. So we're developing what's called a ROCK2 inhibitor, which we believe may provide a multifaceted approach to CCM or angioma treatment. So this is just a brief review for people in the audience. So CCMs are these tangled lesions of abnormal blood vessels that occur in the brain and in the spinal cord. And here are a few images. As Helen mentioned, I'm a radiologist by training, so these are things that I love. On the far left, we have an MR image of a single cavernous angioma. And you can see here there's a large a large area of tangled vessels, and then around that, there's a black halo. That black halo is blood product. So what that means or shows is that there's a central area of abnormal vessels, and then there's blood or blood products oozing out from that lesion. And then in the center here, we see what's called, this is a different type of image, but also of the brain. It's called susceptibility weighted imaging. But what it does is, again, it shows blood products, but blood products that are within a malformation. And so here you can see that both sides of the brain have several of these angioma lesions in them. And this is a person who has a familial form of CCM. And then I just want to pull your attention to the far right here. This is a cartoon or a diagram of what does a CCM look like up close. So CCMs are characterized or commonly referred to as mulberry type lesions. And what happens really in CCM is that the cell-to-cell junctions, the cells that form your vessels, or that form blood vessels, usually have very tight junctions between them. But in CCM, there's difficulty in making a functional tight junction. So you get a looser or not as well formed junction. And then what happens when you put blood or liquid through there, you get some pressure. And if you ever try to squeeze a balloon or you have a weak point in that latex, you know that that's a place you're going to get an outpouching, right? And so what's happening here is that you have several of these inadequate or not well-formed junctions, and then you have internal pressure from the blood, and it's making these outpouchings. And those outpouchings, as you would know, if you've ever popped a balloon like that, is an area where there's more fragility or more weakness or a vulnerable area. And so that's what we're hoping to make less vulnerable with treatment. So moving on to the next slide, a little bit more about CCM. So on the top right, this is a picture of healthy vessels or healthy vasculature. I just want to call your attention to these kind of peach-like cells. These are endothelial cells. It's a big word that really just means the cells that form the blood vessel. They make that tube or that pipe through which the blood can flow. And I want to show you, especially these blue spots in between. Those are tight junctions, representations of tight junctions. So these cells get very, very close together. And that's important because the blood flows then through these tubes or these pipe-like formations. And these tight junctions are what enable the blood to stay inside of these endothelial cells. So that's what a normal vessel looks like. And then we have, the associated neuronal cells. So these are supportive of neurons. So it's an example here. And what I just want to point out is that they're in close proximity to the blood vessels. So the point here is that when something is wrong here, it travels easily also into the adjacent brain. And then we can see in the bottom left, this is an example of what happens in CCM. So the vessels in CCM are not like the vessels in healthy vasculature. And what we can see is that there's a loss of those tight junctions. You no longer see those blue areas holding the cells together. There's some space in between these cells, and we end up seeing red blood cells or blood that was usually contained inside the endothelial cells able to leak out into the adjacent brain or neuronal tissue. This is a problem in CCM, right? So once you get these bloods leaking out, then you also get blood where it doesn't belong. The body responds with inflammation. You can see some inflammatory cells are making their way over to that area. And you can, in fact, even get inflammatory cells coming in through these porous areas in your blood vessels. So really what's happening in CCM vessels as compared to healthy vessels are you're ending up with increased leakiness, an increased likelihood of bleeding. When you have that, you also have inflammation because you've got blood in the wrong place. Your body responds with inflammation. And then the downstream consequence of inflammation is something called fibrosis, which is just an early form of scarring. Eventually will evolve into scarring. So what we're trying to do is to restore a healthy vasculature. So this is just a cartoon here of an analogy. So in normal blood vessels, we haven't gotten to this part yet, but we'll talk about it in just a minute. The ROCK2, this protein activity is very low. ROCK2 happens to be activated primarily in stress states when something is wrong. And so when you have low levels of ROCK2, you have a normal barrier represented here by this sieve. You put things in and very few things come through, right? For example, here, none of the red blood cells are leaking through the functional tissue or the functional sieve. But on the right here, when you have high levels of ROCK2 activity, that barrier function is no longer intact, and you end up with red blood cells able to leak right through. And conversely, actually, inflammatory cells can come right back in. So you're no longer having that functional barrier. That's the main point, is that in CCM lesions, your blood vessels don't provide the same functional barrier that healthy blood vessels do. So what do we do about that? What does CCM management look like? So we know once a lesion has bled, there's a very real risk of another bleed. So we'd like to reduce that risk of the next bleed. There also might be symptoms from that original bleed. We'd like to be able to reduce inflammation and whatever else it might be that is causing those symptoms. And then we know even when symptoms don't persist, the risk of bleed can still impact physical and mental function. So for example, just like Robin was speaking about in the last session, there's a lot of questions for people. Can I go on an airplane? Am I feeling in general just discombobulated? I want to share, I had the opportunity to go virtually to a conference in New Mexico of CCM patients, and there was a man who shared his own journey with CCM. And what struck me about it was just the unpredictability of CCM and its symptoms and how that can affect really career and family planning. So in this case, the man was a fireman, and he was on a call. And while he was on this call, he had his first onset of symptoms. And so he went to the hospital. The hospital diagnosed him, talked a little bit about what his options might be. It turns out that he had a familial form of CCM, and so surgery wasn't maybe the first thing they would want to do. And he went back to work, and within a month or something like that, he had had another bleed. So he was at work, called again to the field in the midst of fighting a fire, and he had an onset of symptoms again. And he said, you know, this is irresponsible of me to be at risk for having these symptoms while I'm trying to be responsive and emergency responder. I don't think this is an appropriate role for me anymore. And so he changed what he was doing in his career. And then it turns out he didn't have another onset of symptoms for 14 years after that. And that just really hit home the unpredictability. So it's not even necessarily the symptoms, but just the risk of something happening that can really change people's lives. And we, as Jeremy said, are very concerned with that community and would really like to be able to contribute to a more normal ability to plan ahead. And so what if we could address symptoms and bleed? What would we look at? So this is where we get into ROCK2. So as Jeremy said earlier, ROCKs are proteins, ROCK1 and ROCK2, and we're particularly interested in ROCK2. And they're proteins that look to be very important in CCM. So what we know is that cells that have faulty CCM1, 2, or 3 proteins, so those are the 3 genes that we know can cause familial forms of CCM. When you have a faulty CCM1, 2, or 3 protein, you end up with very high levels of another protein called RhoA. You can see that down on the bottom left. This is a Western blot. We don't expect anybody to be able to decode this, but what you can see here on the far left is that in normal or wild type, that's the abbreviation cells, levels of RhoA are very low. When you have no CCM1, 2, or 3 in these 3 next columns, you get very high levels of this protein called RhoA. They've quantified it in the next figure here. It looks like it's 20 to 35-fold more. So that's an awful lot more of this RhoA protein. So what does RhoA do, and why is that important? The RhoA lives very near the cell membrane or the border of the cell, and it receives incoming signals, from outside of the cells. And when it gets those signals, then RhoA passes those signals on to ROCK2, and ROCK2 goes ahead, travels throughout the cell, and disperses that signal. And unfortunately, it disperses signals that lead to vascular leakiness, to increase cytokines and inflammation. Cytokines are molecules that can promote inflammation, and it can lead then to fibrosis and scarring. So ROCK2 really is a molecule that tends to be activated in states of stress and in states of disease, and it has this sort of panic reaction where you're getting leakiness, inflammation, and fibrosis. And so we're really looking at that as a target for successful CCM treatment. So one summary here, why is ROCK2 important in CCM? So in CCM, we see increased activation of ROCK2. So how does that happen? Mutations in any of the CCM proteins 1, 2, or 3 actually impair the formation of what's called the CCM complex. So you need all 3 of those proteins to form this complex. This complex inhibits row A. So when you have that together, you have low levels of row A like we saw on the last slide. When you don't have that complex, you get increased row A, increased ROCK2, and that's a problem because that's stress signaling, which causes leaky cell junctions, increased inflammation, and initiates fibrosis, all things that we see in CCM lesions. So what might happen if we could block ROCK2? So what we're hoping is if that we could block ROCK2, stress signaling would be halted or reduced, and the endothelial cells, those vascular cells, could then return to their normal balanced state. Now, if they return to their normal balanced state, then this weakened junction is eliminated. We have a tight junction restored. The red blood cells can stay within the vessels, and the inflammatory cells are never even recruited in the first place, but any that happen to be lingering around can't penetrate through this tight junction, and they're successfully kept outside. What you have is then a normal vasculature without a risk of bleed. So that's the goal. And this is some information from the literature where people really have explored what would it look like if we blocked ROCK2. So this is from Borakova et al. And what they did is they took blood vessel cells or endothelial cells. These are normal cells on the top row. And what they show is that if you leave it alone and put it in a dish and incubate, in about 3 to 15 hours, you get these what are vessel-like formations. They also looked at blood vessel cells that didn't have CCM1, 2, or 3 proteins. In this example here, it's just CCM1, but they did do it for all 3. And what we see here is that no matter how long you leave them to sit there, they do not successfully form those tubes. They don't have those proteins. They can't form those tube-like structures. Now we can see here, they treated with a drug that inhibits a little bit of ROCK1, a little bit of ROCK2. And what we can see is that eventually they do get to some vessel-like tube structures here later on. And this last experiment they did, they directly blocked ROCK2 with something called RNA. But the point here is that they got rid of the ROCK2 protein in these cells. And what we can see is almost normal vessel tube-like formation here, suggesting that ROCK2 really is an important mediator in inhibiting that vessel-like formation and is an excellent therapeutic target. If we move to the next slide, this is work we've done in our own research labs. Like Jeremy said, we are scientists and we are pioneering immunology in drug development. And what we can show here is that when we block ROCK2, we have a potential restoration of immune balance. So we can see decreased inflammation, which is marked by a number of these. We have no expectation that you would recognize any of these names, but these are names of molecules that cause inflammation. And these are names of molecules here that decrease inflammation. And what we can see is that when we treat with ROCK2 inhibitors, we see a dose-dependent decrease in these pro-inflammatory cytokines, or the molecules that can cause inflammation. And we see an increase in molecules that can quell or decrease inflammation. So looks like ROCK2 inhibition can restore immune balance, decrease inflammation. We also looked at how does ROCK2 inhibition affect fibrosis, right? Fibrosis is part of what causes cells to be pulled apart or makes it difficult to form a tight junction. So this is an area of interest here. And what we can see, again, these are very difficult to read. But if you look at the blue bars from the right to the left, which is also an awkward way to read, but just bear with us, you can see they're increasing concentrations from the right to the left. As we increase the concentration of ROCK2 inhibition, we see decreases in all 3 of these markers. And again, no expectation to recognize the names, only to understand that these are markers of fibrosis. And so when we treat with ROCK2 inhibition, we can see that these markers of fibrosis decrease. So just to summarize this slide, we've shown in our own in-house labs that when you treat cells with ROCK2 inhibition, you decrease a pro-inflammatory response, you increase an anti-inflammatory response, and you decrease an ability to cause fibrosis or scarring. So all good things. And here we have an animal experiment from the literature. This is a very important paper by McCarricher et al. And what they did is they took mice, they bred mice who were very vulnerable to developing CCM. So in this case, they're CCM3 mice. And then they looked at what happens if in these mice that are very prone to developing angiomas, what happens if we have decreased ROCK1? And what happens if we have decreased ROCK2? And so what we can see is that if you decrease ROCK1, not much happens. They have similar numbers of lesions. Here, this is the volume or the amount of lesion that stays the same, whether you have a normal level of ROCK1 or on the right, a decreased level of ROCK1. The blood, which is the blue, the amount of blood, is also the same. But if we move over here to the right, where they looked at these same mice who are prone to develop angiomas, and then they decrease the amount of ROCK2, so normal amounts of ROCK2 on the left, decreased amounts of ROCK2 on the right, and what we can see is that really significantly decreases the number of lesions these mice have and very significantly decreases the amount of blood we see from these lesions. So very important. This is just to highlight that ROCK2 really looks like the functional target here for CCM. And fortunately, as Jeremy said, we have a selective ROCK2 inhibitor. It's the OV888 GV101 capsule. This particular capsule has over 1,000-fold selectivity for ROCK2. This is very important. Why are we even talking about this? Because as Jeremy said earlier, there are two proteins that are ROCks, ROCK1 and ROCK2, and they look a lot alike, but they do different things. So it's very difficult to get a drug that only acts on one of them rather than on both of them. But it's important because when you act on both of them, you don't see the same anti-inflammatory effects, and you can be limited by some other side effects that come with blocking ROCK1, which it looks like you don't need to block in this indication or in this disease. And so we're very thrilled to have this special selective ROCK2 inhibitor for development. And we're just going to run through a summary of blocking ROCK2 and CCM. So what can that do? When we block ROCK2, it looks like we can restore the ability to form vessel-like tubes, right? That we saw in those early slides with the endothelial cells. So we can restore that ability to form tubes. It looks like we can decrease the release of inflammatory cytokines or inflammatory molecules like we saw on that second slide. And it looks like we can interrupt that fibrotic process also on that second slide. So all good things that ROCK2 inhibition looks like it can bring to CCM. Now, how does that compare to other therapies that are in development for CCM? Well, first of all, we know that there are no approved pharmacological therapies for CCM at this point. Part of the reason really to get behind it and bring something to this population. Drugs that are being studied at this point are the atorvastatin. Atorvastatin is a statin. They're pretty widely distributed drugs. A lot of people take them for cholesterol. And they were designed for cholesterol inhibition. But also, they have this other mechanism where they can decrease ROCK signaling. So atorvastatin acts through the same mechanism, but it's not its primary action, and therefore you need a very high dose. Propranolol is another drug that's being looked at for CCM. It acts by beta blockade. It's a blood pressure drug. It has been shown successful in some case reports, but larger trials really haven't borne out that benefit yet. So we'll keep looking at that. Right now, we don't know how that drug relates to ROCK2 inhibition. And then REC-994, which is being developed for CCM, acts by reducing superoxide. Now, what is superoxide? Superoxide is an inflammatory molecule, a short-lived inflammatory molecule. It's called a reactive oxygen species, but it buzzes around and causes some of that inflammation that we're talking about. And reducing superoxide may also be one of the effects of ROCK2 inhibition. So there might be some synergistic work there. Now, where are we in development? So this is exciting and important. We are thrilled to share that the OV888 GV101 capsule has already been studied in healthy volunteers at many doses, at doses above what we were thinking would be the target for a CCM trial. We have completed dosing participants. We already know there were no serious adverse events, no high-grade clinical signs or symptoms to worry about. And we look forward to sharing detailed results of this trial when they're available, and we're hoping that that is soon. But importantly, there were no what we called serious adverse events. So where does that leave us? This is a typical clinical development timeline. There's a basic research or drug discovery, which is a lot of that basic science Jeremy was talking about. We've been through that. The preclinical research, animal safety, can we move this into humans? Yes, we're there. Phase 1, we test in healthy volunteers to make sure that it looks safe to advance on to phase 2. So we've completed that phase 1 testing. We're thrilled to share that. And we're working now to get a clinical trial into patients with CCM. After that, we move on to expanded larger trials. Eventually, there's regulatory body review. Should not be just FDA here, but there are a number of different regulatory bodies depending on your country, in this case, MHRA, and eventually approval. But we're here mid-range, and we're very excited to be moving on to trials in patients. And this is the last slide, just to say thanks so much for being here today, for learning about this with us. Again, together we can make a difference. So as Jeremy really spoke so eloquently about, this really is a giant effort by a lot of people. The Alliance to Cure, the Cavernoma Alliance UK, obviously, with Helen, thank you so much. And then with pharma, being able to partner with the people behind the cause and hopefully bring some therapy to this indication. All right, I think that marks the end of my slides here. So maybe Helen, do I just stop sharing then?
Yes, if you could stop sharing, that would be absolutely fantastic.
Excellent. There we go.
Thank you so much, Jeremy, and thank you so much, Amy. That was incredibly informative. And, you know, I'm aware of the drug you're developing, but, you know, I still, each time you talk about it, I'm learning something. And I know it will be of great interest to our members joining today. So we're now going to move to the part of the webinar where we invite questions. So if you'd like to submit a question to Amy or Jeremy, please do put it in the Q&A. Just a reminder, I'm conscious we do have clinicians here. However, they can't give you medical advice on your own condition. They don't have access to your full notes, so they can only talk in general terms. So I can see we've already got one question in the Q&A, so I'm going to read it out and then I'll leave you to decide who's best placed to answer. Thank you very much for your presentation. Just to clarify then, ROCK2 inhibition would only be beneficial for familial CCM, i.e. only when you have the gene mutation, or could it inhibit also for sporadic CCMs? Also, does the siRNA inhibition show better outcomes, and is that where you see the future on the treatment?
Amy, may I start and then you follow? Is that okay?
Absolutely. Go for it. Mm-hmm.
So the way that biology works is really interesting. You can try and target a gene, or you can try and target an RNA, or you can try and target downstream. The RNA makes a protein. That protein then has a function within the cell. Now, it transpires that in both the genetic and also the spontaneous arena, what we are clear on is that the ROCK2, if you knock it down, it will affect both. And that's actually a very important concept because, yes, you're not tackling the gene. That's incredibly difficult. That will be years in the making, and it will not be easy to do. I've tried this in many other disorders, and very few have been successful to date. They will be in the future, not now. So what we've done is to look at the key nexus of all of the proteins that we think are important, and we believe ROCK2 is critical for the shape of the cells, for the way they stick together, If we can knock that down, and we've demonstrated in mice that you can do that and you can make these lesions shrink, then we at least are making a start. Now, that doesn't mean that in 10, 15 years from now, that there won't be a genetic therapy. There may be. I can't speak to that. But don't think of this as being exclusive for one type or the other. The one thing that you should think about is that ROCK2 covers both. Amy?
Oh, that's excellent. And just to add to that, we see in most cases of sporadic CCM, there are mutations in CCM 1, 2, or 3. In almost all. So it still is that protein complex that's causing the issue. So as Jeremy said, the inhibition of ROCK2 is expected to be effective for both populations, both sporadic and familial.
And just to add, as a patient advocacy organisation, something we're really passionate about is treatments being developed that are inclusive of as many people within our community, both familial and sporadic, as well as children and adults. So just to say, that's something we will continually do as we talk to a range of different organisations, advocating for the inclusion of as many people as possible, because we know how important that is to you all. So I'm going to move on to the next question, and just to say, if you've got any other questions you'd like to ask, please do pop them in the Q&A. We love to receive your questions. So this question is, thank you both, very helpful. What is the expected timeline for completing phase 3 testing and approval?
Amy, you want to take a crack at that?
Sure, we'll take that one. So timelines are hard to plan, but what we can say is we're moving ahead with our phase 2 testing. And one thing we need to do in clinical development is take the results from the earlier stage testing and then apply them on to the later stage testing. And then this is a rare disease, and so sometimes rare diseases can go a little bit faster. And there are, as we know, no approved therapies. And so we always hope, we're small biotech, speed and time are very important to us. So we're always active in trying to move the trials along as quickly as possible. I can't give you a date on that. Go ahead.
But let me offer you some thoughts. The community can play a role in this, in speeding this up. It's very important. There are the way a drug is developed requires several key steps. One, a dialogue with the regulatory authorities who say, we believe that that's the right measurable endpoint, an endpoint that is meaningful to the community, to doctors, to the families, to the company. That's number one. So they're in helping us define and working with the regulatory authorities when they've never had the chance to do this. They do not know. I want you to understand, they do not know what this disorder is. So educating them and helping them know what's important is step number one. And we've done that before. We did this in Angelman's, and I'm so proud that we did this. When Ovid started in Angelman's, we educated with the help of the community, the regulatory authorities. We defined an endpoint, and Angelman's, as a consequence, we then were able to run a trial, the first company ever. Now, that was all community-based. We were locked like this, and we had to really, really educate those people at the FDA, EMEA. Gosh, they just knew nothing about these disorders. But when we did, they said, wow, that's clever. You can use that as an endpoint. That was like that with the community. Once we did that, it was an open sesame, because what happened next was that Ovid ran its trial. Its first trial was successful. And frankly, what happened next was that many companies sort of stampeded into the area with the intent to change it using that endpoint in the clinical trial. So that's one, the endpoint and talking to the regulatory authorities. You as a community have a profound role to play in that. The next is equally important. You need to gain trust in us. Why? Because if you don't trust us, you won't work with us. And I know that, and you know that. And there are a lot of people who stand on a soapbox and say, I've got the next best thing from, I don't know what. I actually happen to like a bacon sandwich, but that could be. That being said, the really important thing here is that when we tell the community how we plan to run the trial, we'll be working with you to do that. Then when we say we need to have the right kind of individuals in this trial so we can show the right kind of result, we need two things. One, We're going to need your help to enroll. Absolutely. Absolutely. We're going to need that. That's going to be crucial, but that starts with trusting us. The second thing, we're going to ask your forgiveness right at the beginning, because not everybody can enroll. What you want to, there will be selections of people who fit the criteria of the clinical trial. But once you do that, you will go in and we'll do it as rapidly as we can. And the two things that delay trials more than anything else, is not understanding that you have to get a good endpoint with the FDA and not devoting the time and not working with you. And number two is enrollment. So thank you. It doesn't give you a date, but it gives you a process. And I hope that we absolutely, Helen, you and all of the patient organizations will be working with you.
Well, two very important points there, and we want to thank all of our members through Cavernoma Awareness Month for all that you've done resharing our posts on social media. We reached over 15,000 people through this month, which is extraordinary. And as Jeremy said, that is so key, educating and improving understanding with key stakeholders like the regulators. And in terms of that relationship in between pharma and patient advocacy organisations, we're really appreciative of that opportunity to have that dialogue. And from a patient perspective, we will speak up, share your needs. And we're really appreciative of Ovid and Graviton and taking the time to sit at the table with us and to listen to those needs and to look at how those can be acted on in the development of medications. And as Professor Rustam Al-Shahi-Salman said this morning, if you're eligible, and if you feel able to, enrolling on a research study is so important because it's a rare condition. One of the biggest barriers to getting research off the ground is good enrolment, and that's a difference everybody can make. So we've got 3 minutes before we come to the end of the webinar. I've had a few questions come in, so let's see how many we can get through. Hopefully we can do a couple more. And if we can't get to all of them, I'll make a note and I'll pass them on. So the next question is, how many healthy volunteers were tested in phase 1 and what were the doses?
Absolutely. So in phase 1 testing for the OV888 GB101 capsule, there were 10, 20, 38 people in a 7-day study at doses of 100, 200, 400, and 600. 30 of those people were exposed to the drug. 8 of those people were the placebo controls. There was also a 12-person food effect study. So in total, there are 50 people. And this is getting a little bit more complicated, but the molecule has also been tested in phase 1 setting, in phase 1 studies in a liquid formulation or drinkable formulation. And in that formulation, about another 10, 20, 50, 62 people. And of those 10 were treated with placebo. So all in, about 90 to 100 people have, healthy volunteers have received the study drug. And the 7-day doses have been up to 600 milligrams. And the single doses are double that, up to 1,200 milligrams. And I think I won't say yet exactly what we're going to dose with in our clinical trial, but once we sort that out, we'll let you know. We don't think we're going to need to go to that full top dose.
For those, I don't believe everybody, like myself, you probably have to get educated on clinical trials, and I hope you do. It's very important. And I was struck by how many people are going to hit the web and say, okay, this is great, but need to find out. The numbers that Amy's just mentioned to you are very typical of what you do in a phase 1. In fact, they're quite a lot, they're more actually than you normally have. And some people just do 10, 20 people. We've done a lot.
So thank you so much, Amy and Jeremy, for your answer. So I'm just going to ask one last question before we wrap up, and apologies to those who we've not got to. I will collate those questions and pose them to Jeremy and Amy in email. And that is from people who have spinal cavernomas. We've had a couple of people saying, could this be of benefit to people with spinal cavernomas? May they also be enrolled in trials? So if you could just say a little bit about spinal cavernomas, please.
Amy, shall I take a crack first and then you follow up, okay?
Okay.
So, provided the pathology of the origins of the cavernoma is that, then the likelihood is it will have an effect. We don't know that yet. We're not measuring for that because it's difficult. But don't forget, there's not just those cavernomas. There's also there's ones in the retina as well, which are equally there. So one has to look very carefully at the type of lesion and make sure that the diagnosis is absolutely consistent with what you see in the brain and the brainstem.
Sure. And just to add to that, what we can say about spinal is we did look at including them in clinical trials. We may not, although the treatment should be applicable because the disease process, as we know, is the same. Spinal angiomas there's a very high percentage of them that are surgically removed. So they're very strong candidates for surgery. So it's less likely that people with spinal angiomas will complete the duration of the trial. So for us, that makes it difficult to investigate the drug. And then the other complication, unfortunately, with spinal is the imaging is just much different, and it's hard to put it together in the same trial. So for our initial look, we haven't been able to figure out how to include this group. We know, we are aware, we know people with spinal angiomas are also affected and also would want the ability to participate. So it's something that we have ongoing. Same thing we have ongoing is looking at how can we decrease that lower age limit? How can we increase pediatric participation? So a lot of people we know aren't going to be able to be included initially in our very early studies with patients, but we're looking very actively at how can we broaden the study and be as inclusive as possible as we move forward. So thanks for that question.
I think that's an important point about this as a journey, and it will start with brain cavernoma adults, but it's a journey where you're going to look to then hopefully include as many people as possible as we go along, but it will take time. Thank you so much to you both. It's been an incredibly informative session. We will be keeping in touch with both of you. So for members wondering what next, as we hear more developments, we will keep you updated on our social media pages and also in our newsletter. So thank you ever so much.